I hope phagocytosis is clear to you. So in phagocytosis, what was happening? Your macrophage basically is get attracted by the proteins released by the bacteria, okay, and moves towards it, engulfs it, fuses with the lysosome, forms phagolysosomes, and lysosomes digest it, and nutrients are taken by the macrophage, and rest of it is egested out by exocytosis, isn't it? Now today, as you can see here, it's a complement protein I'm going to talk about, and again I will come back to this phagocytosis also in this part also. So moving on with complement proteins. Now there are basically, as you know, in the first video also I have said there are 20 what complement proteins. Okay. Now where are these proteins? They are present in your blood. Present in blood. In what form? In inactive form. So they are present in your blood in an inactive form, and they are produced by, from where they are being made, they are produced in liver. Okay, so basically these proteins are present in your blood, and there are 20 complement proteins, but they are present in the inactive form in your blood. Basically they act as what? They act as, uh, enhancers of inflammation, okay, or rather you can say they are the substances which enhance inflammation. Okay, so you can also call them as inflammation mediators. What do we call them? Inflammation mediators. So they basically act as inflammation mediators, and also you can, and they are also antibacterial in nature. Okay, they are what? Antibacterial in nature, means they try to act against bacteria, isn't it? Antibacterial in nature. Now the question arises, what, you know, what triggers them? Because they are inactive, what triggers them? So it's basically, you know, that your now bacterial cell wall, you know, it's different, you know. So basically they are able to, they are quite unique. Now bacterial cell wall is made up of what? Murine polysaccharide, you know, and it is unique in nature, which is detected, you know, which is detected now by these proteins, and it triggers them. Your immune system detects it and it triggers your complement system. Okay, so that triggers your these proteins. Okay, basically your bacterial cell wall triggers them. Okay, triggers them. Okay, now how do these complement proteins they act? They act by various ways. See, they are, these complement proteins are involved in both kind of immunity, whether it is innate immunity, like any bacteria entering your, or virus entering your body, they get activated, that is non-specific, non-learned immunity. And when we talk about acquired immunity, that is learned immunity, there is an antigen antibody interaction. So they are involved in both the kinds of immunity, whether it is innate or active. Okay, so time to time I will talk about it in case of active immunity also. Now basically what they do, now how do these they act against these bacterial, na? So basically you see there is a cascade of protein, complement protein means that if one of the complement protein, suppose I say there is a complement protein C1 gets activated, what it does, it activates C2, C3, C4, like that. So it's a cascade of chain reaction, you can say. And in this reaction, what these complement proteins they do, they form a hollow tube like structures. What they form? They form a hollow tube like structures, and suppose there is a bacterial cell around. Suppose there is a bacterial cell around, what they do? They create these tube like structures. They create what? Holes in it. So what they do? They basically, you will see, they will create what? Holes, where they will create holes in the bacterial wall. Huh? They will create holes in the bacterial wall. Once they create the holes in the bacterial wall, what will happen? These cells, these bacterial cell will swell and it will burst later. Okay, so it will swell and it will burst, and it will cause what? Lysis. This is called as lysis of bacterial cell. That is, ultimately they are causing the death of the bacterial cell, isn't it? So bacteria, they will create hole which absorbs takes up water, and it swells and it bursts open it. Okay, this is one way it works. Now there is another kind of a protein which we call as opsonins. What do we call it as? Opsonins. Basically, the presence of infection or bacteria around. You know, I said it stimulates your what? These complement proteins. Now suppose there is a bacteria, and you know that the bacteria on them has got certain antigenic proteins on it surface, which is an antigenic determinants, you know. Now what these now complement protein, there is one of the complement protein which we call as opsonins. What do we call them? Opsonins. Ready to eat. Okay, opsonins. The opsonins, what they do, they attach themselves to these antigenic sites. Okay, to the antigenic sites of the bacteria, to that those proteins on the wall of the bacteria, they attach themselves, these opsonins. Now in the previous video I have talked about these macrophages, if you remember. And there is a macrophage, and this macrophage, what it does, it has on its surface, it has got what? It has got the receptors. It has got the receptors for these opsonins. What it has? It has the receptors for these opsonins. Now opsonins, what they are doing, they are tagging those viruses. Yes, it's a foreign body inside, now you come and eat it. Okay, so what will happen, these opsonins will attach itself. Now suppose I'm just making for you one here, showing you. See, this is the opsonin, and now it gets attached with this bacteria. Okay, now this bacteria will be later on what? Engulfed by this macrophage. It will phagocytize it, because this opsonins receptors by which they attach to it, and later on this entire bacteria will be, as you have learned now, phagocytosis, I need not to tell it again and again. So they will now engulf it and they will eat this bacteria. So basically what your now complement proteins they are doing, just they are acting as what? I have told you people, they are acting as what? Enhances or it complements your what? It enhances inflammation. First point, enhances inflammation. Okay, secondly what it does, attracts phagocytes. I hope it's clear. Like opsonins, and this process is called as opsonization. Opsonization. Okay, and like means ready to eat. Opsonins, Greek, it means that. Okay, and they also call what? And the third we read about, they create holes. So they also act as membrane attack complex. They act as what? Membrane attack complex, which causes basically what? It causes cytolysis. What is cytolysis? That is breakage of your cell. They break open your what? Bacterial cell. That is called as cytolysis. I hope complement system is clear. 20 proteins, inactive proteins present in your blood, and they are what? Basically your complement mediators. Okay, they attract bacteria. They are antibacterial in nature. They attract phagocytes, and they also create a membrane complex, and create holes in the bacteria and causes their lysis. Okay, so see you. Welcome all of you to the next video later. Thank you.